Good evening to one and all present here. Am I audible? Am I audible?
Yes, sir. Yes, sir, you are audible.
Yes, sir. Yes, sir.
Good evening to one and all present here.
Good evening, sir.
Am I audible?
Yes, audible, sir.
No one is responding.
Audible,
Am
sir.
I audible, sir? Anyone? Okay, today is day 5, session 9. The topic is AI in laboratory equipments. The expert is Mr. Sendil Nathan, the Managing Director of Fusion Green Health Care Private Limited, Chennai. On behalf of Jayasakti Engineering College, Management, Principal, Director Academics, Dean Research, Organizing Committee, faculty members, and all participants, we welcome you, sir. Thanks for accepting my invitation and for accepting to deliver this session. I would like to give a small introduction about Dr. Mr. Sendilnathan, sir. Sendilnathan has graduated BE Electrical and Electronics Engineering at Annamalai University in 1999, and sir is having over 21 years of experience in healthcare and medical diagnostics. And sir worked as a area business manager, key account manager, as well as senior area sales manager. Now, sir is the managing director of Fusion Green Healthcare Private Limited, Chennai. Mr. Sendilnathan Manikam is a member of Asian African Chamber of Commerce and Industry and a versatile and accomplished healthcare professional with over 2 decades of experience in the medical diagnostics and healthcare technology sector. Since 2014, he has been serving as a managing director of Fusion Green Healthcare Private Limited, leading mission to deliver innovative diagnostic solutions and sustainable healthcare technologies across India. As a dynamic and result-oriented leader, Mr. Sendilnathan Manikam combined technical expertise, strategic foresight, and business acumen to drive consistent growth and operational experience. His professional journey began in 2003 with Nicholas Pyramid Limited, where he built a strong foundation in sales management, service coordination, and distributor operations. Over the years, he has advanced through he has advanced through key leadership positions at Roche Diagnostics India and Beckman Coulter Diagnostics, gaining deep expertise in clinical diagnostics, business development, and customer relationship management. An electrical and electronics engineering graduate from Annamalai University, he brings up a unique blend of technical depth and strategic leadership to the healthcare domain. Under his guidance, Fusion Green Healthcare has grown into a trusted and progressive organization in India's medical device distribution and diagnostics innovation ecosystem. Recognized for his visionary leadership, commitment to ethics, and focused on innovation, he has continued to inspire excellence in the healthcare industry. He remained dedicated to advancing diagnostics accessibility, strengthening partnerships, and the shaping the future of healthcare delivery through quality, integrity, and sustainable growth. So I once again welcome Mr. Sendilnathan Manikam sir to take over this session. Thank you, sir.
Thank you, Dr. Raja, sir, for giving me such a vibrant and superb introduction about me. It's really motivating
Yes,
me
sir.
again. Is it audible, sir? I'm audible, sir?
Yes, sir,
Hello.
you're clearly audible. We can hear you.
Okay, sir. Thank you, Raja, sir, for giving me such a clear introduction about me, and it really means a lot to me. And without wasting the time, I'm just getting directly into the our presentation. Mainly, I think we had a discussion with
Sir,
Raja sir about.
voice is not audible for me.
So this
Sir.
is, I guess,
Others, it is
Rajas has got a
audible.
problem since start.
For others,
So
it is
maybe
audible.
we can hear you.
Sir, we can hear you, sir.
Is it on? I'm audible to everyone?
Yes, sir.
Yes, sir.
Thank you. Thank you. Okay, sir. I'm just getting straight away into my topic of when we had a discussion with Raja sir about this particular topic in clinical laboratory. Basically, I am sir introduced me a lot about me. I worked in certain segments, like I was a sales manager, then I was a service engineer, then I went into training purpose. Then I started my own company with OEM imports and own manufacturing later steps. So, frankly speaking, A is getting into the clinical laboratory in certain segments. I'm just getting both in the manufacturing as well as the laboratory usages. So, getting to this first point, I think. So, I think I'll start with the basic things in diagnosis. What will be happening? The medical diagnostics is the process of evaluating medical conditions or diseases by analyzing the symptoms, the medical history, test results, imaging test, and okay. Suppose a patient come to the doctor, it will start with physical examination, then will go to blood test, then diagnosis, imaging, and biopsies at the final stage. So this is the first phase of diagnosis when the patient comes to a physician. How the diagnosis will getting more into the total laboratory process. So obviously these are the basic things. Patient goes to the doctor. Doctors, after physical examination, he orders test. The sample will be taken in different tests, depends upon the patient's health physical conditions, like it's about biochemistry or immunology or microbiology or molecular. So these are the basic things they will test. Then sample is sent to a clinical lab, analyze the sample and post results. The clinical lab provides the result to the doctor. The doctor interprets the results. Doctor informs the patient of the test results. Then the diagnosis and treatment decided by the patient and doctor. This is the second TLP process when the physical examination started. Another process. After this thing, the pathologist, clinical lab scientist, and the medical technologist oversee operation, how they are going to see the operations. The sample collection and processing will be done by the phlebotomist who will take the blood. depends upon the test written by the doctors, and they will be taking different tubes, depends upon the test which are done by the doctor. Hematology will be in a lavender tube, that's EDTA. Clinical chemistry in a plain or a gel tube. Immunology, both are same. Microbiology depends upon the patient test like fecal or saliva or nasal things or blood. Microbiological test based on the blood culture, and pathology might be samples in urine, motion, or blood. Toxicology depends upon the people for the therapeutic drug monitoring and other abuses. Abuse test. How the artificial intelligence in clinical laboratory is going to play a major role. So transforming diagnostics through technology, because in next slides I'll be talking about the analytical phases which will be handling in the clinical laboratories. So transforming diagnostics through technology, AI, machine intelligence with mimics human decision makings. This is the major role what we all are seeing in the AI. Plays key role in medical diagnostics and lab automation, enhances accuracy, speed, and efficiency in lab tests. This will be another added value points for us. Hello. 1 minute. I think something went wrong. So role of AI in clinical labs. The clinical labs alone will not depends upon the laboratory related stuffs. It depends upon the manufacturer. For example, if a reagents or the instrument is manufactured, we will test the AI is playing a major role in process failures. Suppose if an instrument is getting tested before the alignments are the particular segments are getting aligned, or they are going to make the manufacturing process. Something is getting failure at the initial stage itself. AI can able to detect and close the things in the initial phase itself. In some of the right now, what we are doing now, we are doing the manufacturing. At the end of the stage, we will go for the QC test. Then only we will come to know there is a problem in the QC is not getting checked. Then they will go and check the whether it's a mechanical failure or it is a fluidics failure or it is electronic failure. Then only we will be able to know. Because of AI in manufacturing, the depends upon the stage 1, stage 2, stage 3 process itself, we can able to identify the detects or defective things, and we can rectify in the initial phase itself, and we don't need to wait up to the QC checks. It is common for instruments as well as for the reagents, calibrators, and controls. So the IVD malfunction will be initially detected by the AI using the algorithms and other things which you program when doing the manufacture. Second, when it comes to the laboratory, selecting the sample size, sample serum indices, and sample collecting time, and everything can be analyzed in the IVD examination failure. It will be connected. In delayed examination results, it's because of the human error or the people who are approving the results. Final, then physician getting the medical diagnosis failure. Predictive and preventive healthcare support depends on the patients. Applications of AI. Disease detection from test results. Actually, the diagnostic support disease detection from the test results is the major thing which when the pre-analytic, analytic, and post-analytic has done, this disease detection will play a major role in test results. It will give you an idea about certain clinical significance, which is not the sample size, the sample analysis, and everything have happened at the time it will take and give you the results. Because in diagnostics, when a sample comes or before starting the experiment, they'll go for quality control on a daily basis, depends upon the enabled aggregation norms. So they will run 3 level of QCs in the instrument in the morning when they start. Depends upon the level 1, level 2, level 3 QCs and their shifts and trends. The instrument will approve the particular test. Suppose in the middleware. Now AI is play a major role in the middleware of all the companies which is having the total automation. So they will create the rule and trend for the particular parameter, which suppose a glucose is giving you the QC is passed at level 1, level 2, level 3 on the particular CVs and SDs. Then only the machine will allow to run the sample of the particular parameters. So this is another good tool in quality control by having a right control over giving the correct result to the patients. Workflow automation. One is sample handling and result validation. In workflow automation, sample handling will come under pre-analytical data. Result validation will come under post-analytical data. Whenever the sample comes, first of all, we need to check the sample volume is correct. It is perfectly centrifuged. Or the patient sample is having any serum indices like the patient is lipemic, itteric, or hemolytic. Like that, sample handling penalty will be handled by A. And result validation, after validation, the results will be correlated depends upon the algorithm which you written. Then only it will make a post-analytical results in a proper way. Another good thing is data management. Correlate test data with diagnosis. So obviously, whenever the validation, now most of the labs are having the doctors will be validating the results. Before the validation, the doctor is having n number of samples of 1,000 to 2,000 samples a day. They want to do that. Then the doctors will create a rule for the particular parameters to auto-validate and do the diagnosis and give you the report. So this is also possible with the bigger labs, which is having a huge workload on their kitty. Predictive analysis. See, this predictive analysis will not be a reportable one, but it will give you a rough idea about the particular disease risk with the parameters which are analyzed in the instruments. For example, AI application examples we can ask is identify error. One is safety and foreseeable hazards. This is one of the major point in AI, which is going to be a major risk, major role in future. Second is access, estimation of risk to patient, risk evaluation. Third one is quality control. Control risk. Risk control is one of the best thing in the AI, which is playing a major role where when the technicians or doctor loaded the sample, when this gives an initial phase itself, it will eradicate most of the things. Then we will check the parameter. If there's any problem with the kit, we need any stability has gone for the particular kit. We need to recalibrate it, or we need to change the kit. Everything will be done on the control risk control. Monitor, production, post-production monitor. See, after all these three got identified, production and analytical phase and post-production post-analytical phase will be the smoother one for everyone. I think in laboratory, I think most of the time we used to have these three columns we used to discuss, the three boxes. One is artificial intelligence revolution the entire laboratory testing workflow from sample receipt from the final reporting. Of course, yes. Still, they are doing a lot of things on this. An AI-driven system enhance accuracy, reduce human error, dramatically improve turnaround times across all phases of laboratory operations. For example, most of the time, we used to, whenever there is a problem, first of all, we used to check the pre-analytical phase. Suppose in what time they collected the sample. Sample was collected properly, or sample is centrifuged properly, labeling of the sample was done, barcoded properly, and if there any problem in identifying the tubes wrongly. So 80% of the problem will be analyzed mostly in the pre-analytical phase. Now, because of this AI-driven robotic automatic repetitive test and including sample handling, sorting and labeling. Suppose they can take the sample is suppose in bigger hospital like CMC Vellore or PSG or KMCH or any other hospital, means they will be having blood collection area. It depends upon the flows, or depends upon the sections will be there. After collecting the test, first the barcode will be generated, then the barcode will be given to the patient. The patient will take in the sample. After that, they will be paste the barcode in the tube. Then the tubes and everything will be tested by the technician. Then they will put it into the pneumatic system. The pneumatic system will collect all the samples and it will throw the pneumatic things into the pre-analytical area, sorting area. So in this pre-analytical area, we have the pre-analytical systems which will be connected with the TLP, total lab automation systems. Then This pre-analytical system will take care of sample handling, like sorting the sample, depends upon the instrument, depends upon the different departments, and it will sort and relabel if needed, even the sample is going for a different instrument or different department. With intelligent flags, unsuitable specimens such as hemolyzed sample. I told you about hemolyzed lipemic. Hemolyzed means the patient sample is completely ruptured and it will have a high hemoglobin content, or lipemic means high lipid profile, like TGL will be in white deposits will be there. Yttric means high bilirubin content. So this will be identified using the color of the tubes, which was identified by the AA using a camera in the prenatal area, and it will eradicate the samples. Suppose if the samples are high for the patient, then the serum indices mode will be automatically on in the analytical phase. So for the excess thing, it will add, depends upon the parameter, the concentration will be adjusted either by sample or by the reagents in the spectrophotometer. In turn, it will give you the exact results. So analytical phase is automated analyzer processing complex data sets rapidly, providing enhanced diagnostic capabilities and pattern recognition that surpasses traditional methods in both speed and accuracy. When the pre-analytical is 100% correct, means analytical phase will not have any problem at all. After that, before AA or interface like an laboratory information system like LAS, this will be in they'll be writing in a note or they'll be writing in the things, typing it wrongly, that will be happen. Here, the analytical phase is over. When the results are ready in the analytical phase, automatically you'll go for auto validation, depends upon the rule suggested by the doctors. So after the analytical phase, the rules has been set and will go to the post-analytical phase. AA powered system deliver automated result interpretation, validation, and reporting. This intelligent platform efficiently manage vast data volumes while identifying critical results requiring immediate clinical attention. See, suppose when the in the analytical phase, after results are out and it's going to the post-analytical phase, and when the rule was set by the doctor, depends upon the parameters, and it is perfectly fine, means it no need to wait for anyone. It will auto-validate and will go to the patient and give you the risk-related stuffs. And critically, if the patient has to be immediately addressed, it will be done. Otherwise, if you give the morning the sample, everything will come and the validation and everything only come in the evening. Nowadays, the TAT has been improved. Turnaround time has been improved for patient results. Within 3 to 4 hours, the results are out. Because of this, AA in clinical laboratory. As I always say, AI-driven robotics in pre-analytical systems. If you see the system, this is the system, the standalone pre-analytical system, it's not connected to the track. So this system, it is from Roche because I love this product line. The Cobas P500 represents breakthrough pre-analytical automation, combining robotics and artificial intelligence random sample processing, of course. See, when you see the system, after taking the result, tubes come from the pneumatic system or from the technician's table, you can put it in the basket. The basket will sort and allot and given to the particular system. Then this system will intelligent sample identification will come. Then it depends upon the instrument, the racks, it will align. Then certification, it will go for the certificate, automatic certification will go. Then quality assessment using visual recognition of algorithm. I told about the color, which was color of the system, color of the tube, and color of the sample will be analyzed in the algorithm written from the camera, which is loaded in the clinical system. Smart decapping repack. Automatically it will recap the system, takes allot the sample, and it will again recap the system. Then integrate monitoring with real-time flagging. See, after this, it will go to integrating monitoring with real-time flagging. Then depends upon the instrument rack, it will put and it will go off. By this system, when the workload is heavy, like CMC Vellore and other people who are handling more than 10,000 to 20,000 tubes, which is coming from close to around 562 wards, using pneumatic system, instead of the people going and collecting the sample and sorting the thing, it will take more than 8 to 9 hours. But it'll have within using pneumatic connected with AA and pre-analytical instrument, it will happen only in 30 minutes. You know, where 9 hours come down to 30 minutes is one of the best things because of the this AI-driven robotics with pre-analytical systems. I told the second phase is AI-driven analytical system because mostly the AI-driven analytical systems is useful in digital pathology, imaging, and microbiology. Biochemistry, immunology, yes, after the sample results, because the sample and reagent and sample mixer, either in biochemistry it is spectrophotometer, and in immunology either it is chemiluminescence or fluorescence immunoassay or e-CLIA, depends upon the things. And there is no automation involved in those aspects. After the results only we can able to analyze. In digital pathology, this is making a big impact, very big impact, because most of the time the doctors used to maximum doctor can able to report close to around 60 to 70 slides a day, because viewing a microscope and depending upon the different cells, either in clinical pathology or histopathology or oncology, oncology, clinical pathology, everything put together, it is very difficult for them to analyze everything and do it on manual mode. Now the people, a lot of images can come and they can able to analyze the rules and algorithm set by the pathologist and can give you an idea what is the rough things. After that, it will report and give you the final this thing. So transforming histopathology analysis now become a big thing. Suppose, for example, the customer is having a sample of histopathology. They did the grossing, everything, the block is ready and the slide is ready. It will then they can scan the slide in a system. They can send this scan system the slide to the doctor within minutes, and the doctor can be able to this because it will, when they scan the slide means depends upon the biopsy slides. If single biopsy means it will go up to 3 slides. Large biopsy means it will go up to 20 slides. Everything it will scan and send it to them. It will be easy for the pathologist to view the, so open their software, that particular app user ID which is given by the particular company on this algorithm. They can able to visit and they can able to report within hours. Previously, what we will do, we will take the biopsy, we will do grossings, then staining, a lot of other things, then decapping, and after the slides, they have to put the staining, grossing, everything will go to the, then they courier it to the particular doctor. It will take 2 days time. Then they will see the slide, depends upon the slide which was the biopsy which was sent by the people properly or not. Then they will report after 3 days. So if for a biopsy, after the operation, it takes 8 to 9 days time. Now it is taking in terms of hours. After taking the thing, they will put the slide in the particular platform. It will take the particular platform will screen and send all the data to the doctor, and doctor will report on the same day itself. Then they can go for, then they take a decision on the patient whether they go for surgery or not, not needed or not needed. It will be, the treatment will be very fine. Not, we no need to wait for 10 to 15 days because of the biopsy samples. Now AI-driven analytical hematology and blood analysis. Previously, we used to have a 3-part system which is working on Coulter's principle. They will tell it's impedance technology. The cell will be measured in the 3-part cell counter in terms of size. Size of the cells will be like, it is, if it is large, then it will be WBC. Small, then it will be the RBC. If it is a different size, it will go to platelet. So it will analyze the cell in internal structures. Now after that, the automation came in cell counter. Then dye has come. Then laser has come. So after that, it only to show the differentials. Rest of everything will be done in impedance only. Now after this thing, cell counter, then they come out with the slates and the chain maker. Now they come out with the morphology unit. The morphology unit is like a AI. After the CBC has done, it will, if needed, abnormalities there, it will go for a slide. Then it will prepare the slide automatically. And then the slide will go to the morphology unit, which connected to the AI with the rules. Then automatically it will give you the results, depends upon the structure which was scanned in the morphological unit, which is assigned with the structure and the rules done by a pathologist. So automated cells classification. Then second is morphology analysis. Third one, rare cell detection. Fourth one, quality assurance. These are the process which is making the CBC in a simple way. Otherwise, if with this thing, it takes 2 days time. It is getting closed in 2 to 3 years time. If you have any questions, please raise. I think I'm ready to answer the things, because in some way I'm understanding in some other things in some other way. And if you're not understanding, please let me know. And I already dropped this presentation to sir. So if you want, you can use it. Artificial intelligence for microbiology. See, artificial intelligence revolutions the because one of the in the general labs, people will have biochemistry, immunology, basic clinical pathology. That will be run on the things. Because post-COVID, only microbiology is getting a bigger attention. So now people are getting into microbiology because most of the time, whether the sample is any bacterial infection or microbial infection, we have to analyze. And we have to have minimum 350 square feet for this microbiology setup. And most of the places, they don't have the microbiology department. They have only the chemical things and they will outsource the microbiologists. But here, AI is going, will bring microbiology close to a doorstep with a lot of other steps. Automated colony recognition is one of the best things. AI system analysis, culture plate images, identifying bacterial colonies, and quantifying growth patterns with precision. Resistant direction. So now I think instead of going for the blood culture or this um, blood broth or AST, now the multi-top-like instrument has come with pre-assigned mass spectrophotometric things aligned with AI. So machine learning algorithm predict microbial resistance based on growth, inhibition, zones, and phenometric characteristics, and rapid turnaround times. Previously, we need to wait for culture. They have to wait for first they load the blood culture. It will go show positive, negative in 4 hours time. After that, they have to wait for 4 days. Now this multi-top like automated mass spectrophotometric AI comes. The results will be fine in. It will be done in half an hour time. But slowly it is coming because of the initial investment cost of close to 1.5 crores. Artificial intelligence for genomics and molecular diagnostics. I think the word genomics and molecular diagnostics is already born with AI in only. It is not a separate one. So I think when you're getting into this genomics and molecular diagnostics, the science is getting more involved because I'm also not a great science person. Mostly I'm an engineering with knowing basics of science only. So I'm talking about genomics and molecular. In my sales point of view and my experience, I'm putting some of the words. Unlocking the genomics potentials. AI algorithm interpret vast amount of genomic sequencing data to identify genetic markers associated with the disease, protein structures, and personalized treatment responses. This will be the future because most of the time when the actually molecular become a big role when again the post-COVID only molecular department was known to everyone. Molecular microbiology will play a major role after post-COVID only. So now this sequencing, when you're talking about sequencing, when we see previously before COVID, we used to have in Pan India, we have close to 200 molecular labs and genomics. Now it has gone up to into it doubles, not double. It is 100 times. It has been multiplied, and we have good molecular and genomic labs coming up in a lot of places in India. Particularly, we have good numbers of people. One in Coimbatore, we have Metal Lab, we have Anderson Diagnostics and Genomics based in Chennai, and we have with Genomics Bangalore. So Lifestyle has come out with genomics setup. So lot of genomics and molecular diagnostics came up now, and this is going to be the future. And I told you in the initial stage, genomics and molecular are born with AI only. Without the data and analyzing, we cannot handle those things. And key application, variant interpretation, AI classifies genetic variants as pathogenic, benign, or uncertain significance. Disease prediction, polygenic risk scores calculated from multiple genetic markers. Genomics is one of the predicting drug metabolism, optimizing dosing based on genetic profiles. We have 3 setups in Tamil Nadu. Oncology, precision medicine, identifying targetable mutations and treatment strategies. This is now followed most of the places. Previously, oncology, we used to go only with the biopsies. Now, the markers, the mutations of the cell things comes in the molecular and genomics and plays a major role in oncology, which is enhancing the results in a faster way. The treatments are going in a better way. A lot of patients got rectified. Artificial intelligence for quality control and predictive maintenance. In the initial stages, we talked about how the quality control and the predictive maintenance will make the daily lab life of a technician and doctors in an easier way. Nothing but AI continuously monitor the instrument performance and automatically flag deviations or potential quality issues. When they run the quality control in the morning itself, this will really clearly indicate you the trend. Suppose Yesterday, the is going towards 1 SD or 2 SD or 3 SD. I think trend in upward or trend in lowerward will be predicted in the morning QC itself. Or if you want to go deep into the analysis of the parameter, you can see the real-time monitor of the parameters itself. For example, a particular patient sample is done, we can able to see the real-time monitor, and that can be easily correlated with the morning QC result and can give you light indication whether it is right or wrong. So real-time monitoring of quality control will allow us stating that we can able to provide the correct result to the patient. Then predictive analytics, machine learning model analysis performance patterns and to anticipate component failures and schedule proactive maintenance. Most of the time, I think recently only now the customers are being an engineer. They I think if you see about the Western countries, engineer used to visit the laboratory or hospital once in 2 years, only for the preventive maintenance. But in India, making the customer to understand about the preventive maintenance is the biggest task for the engineers. They will buy the instrument, but they never maintain the instrument. Now only in last 3 years only, the preventive maintenance things are getting, because when a lot of biomedical engineers are coming into the structure, then they will have to take, because most of the biomedical engineers, they're very much happy with the basic imaging instruments and the bedside patient monitor support and life support system they used to see. They never come to the labs. So now the biomedical engineers coming to lab. So they are making this protocol about the maintenance of weekly, daily, bi-weekly, monthly. So now only the customers are now understanding about the importance of the preventive maintenance and the things. So with this last 3 years, the breakdowns of the instrument has come down because of the periodical maintenance. What the biomedical engineers are getting to the lab and they're doing it properly with the support of the technician and the doctors. So the predictive analytics is done by it has to be done, but it is when that is not done, the algorithm what we can write on the system depends upon, like in your car, if you're driving the system, if the timing belt is crossing more than 40,000 kilometers, means it will give an indication that you have to change the thing. Like that, what the AI will do, 40,000 tests run by the system. The PM is not done, the instrument will automatically stop the instrument. Stop the instrument. Once you've done the PM, then you clear everything and click the rule only, then you'll make the instrument to run. That also come into the AI. But most of the time, it is disabled mode only. So, hello? Unable to understand, madam. Okay, let me proceed. So disabled mode only in India. So probably means